Hello students, this is my part 4 video of genetics class 12. So today we will discuss the phenomenon or the cross that is called test cross. Test cross. So what do you mean by test cross? So test means to examine or to know what is the genotype of the organism. So in the cross we have to cross between the organism with double recessive parent. We will discuss what is it. Then this organism, and this question arised because when a tall plant is there, so the tall may be due to capital T. This is homozygous tall. And may be due to capital T small t, heterozygous tall, or we can tell the hybrid. So from this it is very clear that it is very difficult to examine the genotype of the species or the organism without bringing about the cross. So we can do here, this is monohybrid test cross. So tall plant is taken, this capital T small t, and we should take the dwarf one, that is homozygous double recessive, we can also write double recessive, that is small t and small t. So this is Pu1. Then there is gamete formation is there, gametes. So this will produce capital T small t, and this will produce small t. Then when random combination is there, that is fertilization. So these gamete may fuse with this may fuse with this may fuse with this may fuse with this. So this is capital T small t, and this is capital T small t, and this is small t. So this is tall, this is dwarf, and this is dwarf. So this is the F1, and the phenotypic ratio is 2:2, that is tall is to dwarf, or 1:1. So this 1:1 is called as the monohybrid test cross ratio. So when we will get 1:1, we must confirm that this parent what we have taken, and we have to check the genotype. For that we have taken this tall one, whether it is pure tall or homozygous tall or the heterozygous tall. If we will take the homozygous tall, we could not get this ratio, which confirms that when we will get 1:1, then this must be the heterozygous tall or the hybrid. So we will finally conclude this is the hybrid. So suppose we will take this one homozygous tall and bring about the cross, then it is very prominent. You can see here also. Suppose P1 capital T we have taken, and this is double recessive parent, that is dwarf, small t, this is tall, and there is gametogenesis, gametes are formed. So this is capital T, small t. So when fertilization occur, this will cross with this may cross with this may cross with this may cross with this. So they results into capital T small t, capital T small t, capital T small t, capital T small t. So the 100% plant become the tall. This is the tall. So this confirms that if we will take the tall plant and we do not know what is the genotype of this tall, so we have to do the cross with the double recessive parent, and if we will get 1:1 ratio, then we will confirm that this is a hybrid. But when we will take that plant without knowing the genotype, and if it is homozygous tall, then it will always result into 100% tall plants. So this is called monohybrid test cross, and then we will discuss the dihybrid test cross. Then dihybrid test cross. Dihybrid test cross. So similar to that of your monohybrid test cross, we can take here the F1, that is tall, round, and we are going to test whether this F1 is hybrid or pure tall. So we can take as it male one and double recessive parent, that is dwarf, with wrinkle seed, that is small t, smaller. So then gametes are formed. So we can find here 4 different types of gametes, what we have already discussed in dihybrid cross, that is capital T, capital R, then capital T, small t, capital R, small t, small r, and this will produce only one type of gametes, that is TR. So we can make this type of checkerboard also. So these gametes here, and in this way, so this will fuse with this to produce capital T, small t, capital R, small r, then this is capital T, small t, small r, this is small t, capital R, small r, and this is small t, smaller, smaller. So here we will find this is tall, round, this is tall, wrinkle, this is dwarf, round, and this is dwarf, wrinkle. So this is the F1, and in this cross we are getting 1:1:1:1:1. So the dihybrid test cross ratio is 1:1:1:1. So from this ratio, from monohybrid to that of your dihybrid, and then we can also get the polyhybrid test cross ratio also by the simple formula that monohybrid test cross ratio, monohybrid test cross ratio is 1:1. Dihybrid test cross ratio is 1:1 to the power square, or we can write 1:1 into 1:1. Like that we can also get trihybrid test cross ratio 1:1 to the power 3, or we can write 1:1 1:1 1:1. And in polyhybrid test cross ratio is like also 1:1 to the power n. So this test cross is a very important cross in which we can find out the genotype, or whether a plant is hybrid in that respective character or not. So if it is hybrid, it must produce the test cross ratio like monohybrid 1:1, dihybrid 1:1:1, and trihybrid 1:1 to the power 3, and polyhybrid 1:1 to the power n.